Hi, my name is Trix and I'm going to talk about trifluoroacetic acid, or TFA, a type of PFAS that's becoming a growing environmental concern. TFA has the formula CF3COOH. It's a very strong acid, and its 3 fluorine atoms make it extremely stable. In water, it quickly forms the trifluoroacetate anion, CF3COO, which is the main form found in the environment. TFA is mainly produced in the environment from human activities. A major source is the atmospheric breakdown of hydrofluorocarbons, HFCs, used in refrigerants and propellants, which degrade in the air and form TFA as a stable end product. TFA is also generated when pharmaceuticals and pesticides with trifluoromethyl groups are exposed to sunlight and reactive oxygen species in water, leading to their transformation into TFA. Additionally, factories that manufacture or use fluorinated chemicals can release TFA directly into the environment. Nearly all TFA found in nature comes from these human-related sources. TFA is highly mobile in water and does not stick to soils or sediments. It travels easily through rivers, lakes, and groundwater, eventually reaching oceans, and resists natural breakdown. At current environmental levels, TFA is considered to have low toxicity for humans and aquatic life. Some effects are seen at much higher exposures, but these are not typical in the environment. Because TFA is so persistent, ongoing monitoring is recommended. TFA is highly recalcitrant because its chemical structure contains 3 carbon-fluorine bonds, which are among the strongest bonds in organic chemistry. This exceptional bond strength makes TFA extremely resistant to attack by both natural processes, such as microbial degradation or sunlight, and even by advanced water treatment technologies. As a result, TFA remains stable and persistent in the environment, with very few pathways available for its breakdown or removal. Let's look at what happens to TFA in environments without oxygen, such as deep groundwater, sediments, or some treatment systems. Because TFA is already highly recalcitrant, scientists have investigated whether it can break down under these oxygen-free, or anaerobic, conditions. I'll share what research has found about TFA's persistence in these settings. In 1994, researchers investigated whether TFA could be broken down in oxygen-free environments such as sediments and soils. They reported some evidence of TFA degradation and fluoride release, suggesting that certain microbes might be able to transform TFA even without oxygen. Although these findings were intriguing and sparked further research, later studies were unable to reproduce the same results. Still, this early work was important because it opened up new questions about TFA's persistence and possible removal in natural settings. In 1997, researchers looked at whether TFA could break down in environments without oxygen, like anaerobic digesters, sediments, and rumen fluid. They found that TFA stayed stable, with no clear signs of degradation or defluorination, even after a long time. Microbial processes, such as methane production, continued as usual. These results suggested that TFA is very resistant to breaking down in oxygen-free conditions. Kim et al., 2000, ran long-term anaerobic reactors with ethanol-digesting microbes to test TFA breakdown. At low TFA concentrations, they saw partial defluorination. Less than 5% of TFA was degraded, with fluoride ions and small breakdown products detected, showing some cometabolic activity. But when TFA levels increased, methane production stopped and TFA accumulated, suggesting toxicity to the microbes. This study showed that under controlled conditions, only a small fraction of TFA, less than 5%, can be broken down anaerobically, confirming its strong resistance to degradation, especially at higher concentrations. In 2018, Alexandrino et al. Tested the anaerobic degradation of TFA in several real-world samples, including activated sludge, contaminated sediments, and rhizosphere soils. They added TFA to these samples and incubated them under anaerobic conditions for up to 180 days. Throughout the experiment, they monitored TFA concentrations and measured fluoride ion release as an indicator of defluorination. The results were clear: there was no measurable decrease in TFA concentration and no fluoride detected in any of the samples after 6 months. This fact-based study demonstrated that, under realistic environmental conditions, TFA remains extremely persistent and does not undergo anaerobic degradation. Tang et al., 2024, studied TFA degradation using aquifer solids from a contaminated site, kept in anoxic conditions for 10 months. They observed about 3.5% defluorination, with some fluoride released. TFA exposure also reduced microbial diversity and shifted the community, with rare microbes like Flexilinea and Sphingomonas linked to the small breakdown. While some chemical changes were detected, TFA remained mostly resistant to degradation under these realistic, oxygen-free conditions. In summary, decades of research from early laboratory studies to recent real-world experiments show that TFA is extremely resistant to breakdown, especially in oxygen-free environments. Even with advanced monitoring and long-term incubation, only a tiny fraction of TFA is ever degraded, and its presence can disrupt natural microbial communities. As a result, TFA continues to accumulate in water and soil, raising concerns about its long-term environmental impact. These findings highlight the urgent need for continued research, better monitoring, and innovative solutions to address the growing challenge of TFA contamination.